Now let's look at some of the prokaryotic domain in this mini lecture. This is not very long. There are certain groups of bacteria. The gram-negative are called as gracilicutes. Gram-positive are the firmicutes. The ones that are mycoplasm, they are called as the dendricutes. And the archaeobacteria are under Gracilicutes. So I just want you to get familiar with these terms one time. Let's talk about species and strain. What are species? Species are cells that have similar pattern or traits. Within a species you can have different strains. So some species have different characteristics. So sometimes we will talk about different strains. For example, E. coli with a different strain. So they're the same species, but they have some special characteristic. And because of the strain types, it becomes difficult to combat those different species. And the process of serotyping is a process in which unique antibody will be formed according to a response in the host. Some unusual bacteria, for example, Rickettsia, it's a parasitic, it's a gram-negative, and it requires an arthropod as a vector and causes Q fever and RMS. Chlamydias are also unusual bacteria. They're parasitic, do not need a vector, and they are transported through sexually transmitted diseases. And the last one are the Gracilicutes, and these are the ones that are archaeas. They are the ones that are extreme thermophiles. They have no PPG. They have 70S ribosomes, and they also are present on shores of various beaches and oceans, and especially where you have extreme climates, so you won't see them in nice beaches. It's mainly when you have salt-loving bacteria. For example, like the Dead Sea. That's where you will find these, or you would find them in very hot water or very hot springy water. So this was a really brief lecture on this one. Not too much, just a few terms to go over and to review these terms.